does start at 4, right?
It does, yeah.
Usually they are all upstairs chatting, like if one of the masters talks, usually chat with the speaker and call for, and then they come down for reading the last second.
Oh, okay.
And she is here. I just came from that. So
Okay. Yeah.
so don't worry, you're in the right spot.
Yeah. Better be.
Yeah, and then we'll come down for like 5 minutes. Oh, or now.
We're gonna.
It is.
They're all great students. Which is why I feel
Okay, so
anecdotally, it's like that there. I have it's not like I know which things were this work. It's just like, but you're not seeing me like big differences. So there's like different things. We're like really like
Josie,
So just I'd like to there so you can kind of get an idea of like where you need to stand when you put introductions. Sorry, John, I'll say something to you. So if you can just up that face there so you can keep an eye on where you are.
Okay.
So if you don't mind just staying.
Go to the speed stream, that would be where like the food is much more.
And we are now live, so.
Yeah, exactly. Even just like the mountain, like the snakes up on the mountain. Right? But it's just like fancy work too. It's so hard to get samples. I don't know.
Depends on the season.
Like midsummer.
Midsummer.
The last one worked so well. We figured out the production quality of
It's like same as there. It's just it's hard to get a lot.
Man, good stuff.
Yeah, the sprinkles one. Christmas.
Yeah.
Christmas.
They're not the good ones.
Oh, it's the worst color.
The cruelly. Enjoy that.
So, speaking of recording, that conversation was not recording.
I thought everything you were saying was.
He's just Josh again.
By the way, no, that was on the record, right?
My media training was like,
So, okay, we're now live for reals.
Did he? How many did you get right? Is that a 5? Right?
He goes 3. So.
We're at 5.
No, you got 4. You got 4
At least definitely.
That's what
Better get ready for the nice old carnivore.
Yeah, you're gonna get squirrel questions. So you're gonna be
All the grads
and
should
just
get
stupid
it.
questions. Like, which of these is not a famous weather predicting environment?
Did you hear about Jeffrey's questions?
Yeah.
One of them was what's the snake in jungle book?
Cop.
And it wasn't caught. It was like a different snake, but it was like way harder. No one
Yeah,
a snake
in
jungle book. I mean, there's a picture of it on the presentation, so like I don't know. So yeah, that was one of the questions. She was like, "Fine."
No idea. There were multiple choices to be clear.
Oh, really?
That's gonna be better. I'm just trying to
Which one?
not stand.
Who wants to be a millionaire?
Like if I'm standing here, it feels.
So you should do that, and then call me back.
Is anyone gonna be on?
Well, yes. I mean, I don't know.
If I told them like something like call right or ask the audience, we'd probably be like, oh yeah.
Or is that us?
maybe that's.
Oh, no, 2. I think that means someone 10 years younger than me. Right away.
That would be hilarious.
So we have, we've started a project.
Will you do that?
Oh, cool. I also do agroforestry.
There you go.
Well, like pest control.
I think we're thinking that like payment is something how I think people look.
See, I was going to say this is great, $30 a lot, 10 meters.
Oh, man.
20 meters. Yeah.
So
So you're gonna your face this way. So
But you're double dipping
second
that part?
edition.
It's not just like, it looks like it's nice.
Our focus is.
We haven't even started the application yet.
Two. Self-edition. Yeah, I'll wait. Oh, that's what you're allowed.
She's.
No,
she's.
Throw your head back to the wall.
No.
We're also looking at.
As long as you read that.
It'd be really interesting to see how the viewers respond to that. So, they're visiting.
Yeah, I would ask them all.
So we're building awareness, database of our experts in this area. And I don't want to do it, but so.
I was telling her, so I didn't get into detail.
Just so you're-
You sounded a little bit conceptually.
I'll try to different soil, not but yourself.
Yeah.
Even her growth. I didn't say facial lines,
That's just the second semester of the course.
So it sounds like. Yeah, exactly. I don't know what I don't know if it's really
Oh, Montreal.
Okay.
It's changed room because that was that theater.
Yeah.
Someone like a
Yeah,
student.
that was Montreal.
It's very
Let's
like.
get going.
Wow, it's online.
All right, folks, let's get going here. This is our second to last speaker in our environmental science seminar talk. I'm Mark Fraser. We are live streaming. This is our second live streaming event. And it worked last week. I think we had a few sort of issues with the link, but that's been cleared up now, I believe. So it's getting better and better, and Adam is the guy that is making this all happen. So thanks. And now Brandon too. So a huge thank you on that. This talk is also being co-sponsored by the Grassland Conservation Council. And before I introduce our speaker, though, I do want to remind you that next week is our last talk. And our speaker is Lisa Babington from the University of Otago, although I think she's in Alberta right now. And she's going to be giving a fascinating talk titled Testosterone Rules on Science, Fairness, and the Problem with Slowing Women Down. There's been controversies around gender issues, especially in Olympic sports, and I think she'll tackle some of those issues from an ethical standpoint. So it's my pleasure to introduce Chandra Moffat.
Thanks for having me.
Yes, Chandra earned her Master's of Science just down the road at UBC with Jason Pither. And that's where I got to know Chandra, because she got crazily involved in one of our big projects. And she went from her master's to her PhD, where she pursued that at University of New Brunswick. And she started with Agriculture and Agri-Food Canada at the University of New Brunswick. And she's now come back to BC, of course, and is a research scientist at the Summerland Research Station. Now, before I sort of hand it over, I just want to give you a calendar. This is a 2019 calendar. It's put out by the Grassland Conservation Council, and the GCC is co-hosting this seminar. And these are $15 for a calendar, and there's beautiful pictures in it. But we're going to give this to Chandra.
Thank you very much.
Great. So please join me in welcoming Chandra.
So it's
Come talk to
really.
me if you want one of those.
I also have one for sale. They're only $25. Yeah, it's really fun to be here. So today I'm going to talk to you about some work that I did first as a master's student and I've continued collaborating on. And it's on developing a biocontrol program for invasive hawkweeds. And so I'm going to talk to you today about how we combined ecological life history and molecular data to aid in the development of this biocontrol program. So as Locke said, I'm a research scientist with Agri-Food Canada in Summerland. As I was telling the grad students, we're a research center of 27 different science teams. We do all sorts of different things related to sustainable crop production, and we love having people come visit. Just give us 48 hours notice. So before I get started, I just wanted to start with acknowledgements. So this work is work that myself and Dave Ensing, who Locke knows very well, worked on largely together. Dave was first my summer student, and then we were both master's students together and have continued collaborating. And the work is funded by FLNRO and my Tax Accelerate internship grants for myself and Dave, as well as UC Okanagan, and an institute in Switzerland called CABI that I'll talk a little bit about. So this is a field of different invasive hawkweeds just before Wells Gray Park. And when we look on the landscape at distributions of invasive species, we can start to think about why they might be there and what sort of hypotheses that we have in invasion biology to explain the success of invaders. So this is an area where cattle would often graze, even though you can't tell it from the timing of this photo. But maybe invasive species are so successful here because it's a very highly disturbed environment. Maybe these invasive species that have come from Europe have novel weapons, such as allele chemicals or associations with particular mycorrhizae that make them better competitors in their environments. But one of the longest standing hypotheses about the success of invasive plants is that their success is largely due to escaping the natural enemies in their native range that normally would regulate their populations. So this paper was published in 2002. It's very iconic in the field of invasion biology. But this idea that invasive species are successful, at least in part, because of escape from their natural enemies, has its origins much earlier in the scientific literature. So Moran and Vila published a paper just the year before that's only cited about 1/10 as much as Keen and Crowley's paper that presented these same ideas. Gillette in the 1960s presented these ideas. Charles Elton and even Charles Darwin had the concept that invasive species may be successful because of escape from regulation by natural enemies. So we've had these ideas about the success of invasive species for going on almost 200 years, but we're still testing sort of the predictions of these hypotheses today. So in the field of classical biological control, which is some of the work that I do, we aim to harness this hypothesis about invasive species being so successful because of escape from their natural enemies by reuniting invasive pests with specialized natural enemies. So here, if we have an invasive plant species that's been introduced into a new environment, may be very successful, it may start oscillating. But if we do the research to bring in a biological control agent, this specialized natural enemy, the aim is that over time, that biocontrol agent won't eliminate the population of the invasive species that it's designed to control, but will regulate its population so that it becomes much less dense in the environment, it's spreading more slowly, and is below whatever sort of damage threshold we would consider, whether that's ecological or economic sort of damage threshold. And that sort of threshold would depend on the type of species that we're concerned with. So in the biological control of invasive weeds, generally we think about finding one insect species to control one invasive weed species. And this really sort of capitalizes on using very specialized natural enemies or specialized insects. But here in Western North America, we have a complex of invasive weeds called the invasive hawkweeds. And in general, they're thought of in terms of management purposes as being orange and yellow hawkweed. But you can see from these beautiful drawings that I have shown here that there's actually a number of different species within this complex. So we have the orange hawkweed, Pilicella aurantica, and a number of different yellow flowered species. Here we can see them sort of in the flesh, what these species would look like. And just at the sort of first outset, you can get a sense from some of these photos, the density that these invasive hawkweeds can get to. So these are species of Central European origin that have been introduced to Western North America and other parts of North America, as well as New Zealand and South America, largely in contaminant of seed for hay production. And these species, once introduced to Western North America, have become extremely widely dispersed. So this is an old map, and I use this because this is the sort of map that we had access to when I started this work. So this is a map from eFlora in 2010, and it's the distribution of yellow flowered hawkweed species across the province of BC. So these are just the areas, of course, that had been surveyed at that time. So just because there's an absence of a dot on the map doesn't mean there's necessarily an absence of hawkweed. But like many invasive species, the distribution tends to follow major transportation corridors or into other disturbed areas. So the invasion of different yellow and orange flowered hawkweed species in British Columbia has impacted rangeland, reforestation, and natural areas, to the point that the ecological and socioeconomic impacts were forecasted in 2013 to reach up to $60 million a year by 2020, which is in just 2 years from now. So it'd be really interesting to sort of go back to those original data and look at now if they are having those sorts of combined economic and socio-ecological impacts. But we know on the landscape, and I know firsthand from all the field work and surveying work that I've done, that these are extremely pervasive weeds that have direct impacts in reducing forage value, impacting ranchers, impacting reforestation, and outcompeting native flora in natural and protected areas. This is why we want to control them. So as I mentioned a few minutes ago, when we think about controlling invasive species, we really like to think about reuniting them with this specialized natural enemy in this very tightly co-evolved relationship. But when we're dealing with a complex of invasive weeds like I've shown you here, it would probably be a lot more efficient to find one insect that would control multiple invasive species in an invasive complex. So that's what we set out to do. So before I get into a little bit into this, so there was a biological control program for invasive hawkweeds in New Zealand that went on in the 1990s. And there were a number of insects that were screened as part of that. And this just sort of relates to what I was talking with the grad students about. So in New Zealand, there's no native hawkweeds. So there was a number of insects from Europe, from the native range of hawkweeds that could be brought to New Zealand and released because it was considered safe to do so because there weren't native hawkweeds. So in British Columbia, North America, and South America, we do have native hawkweeds. They're in different genera than the invasive hawkweeds that I'm talking to you about. But it meant that we had to look for even more specialized insects because we wouldn't want them to have any non-target impacts on native hawkweeds. So this is sort of where my work came in at this point in time. And the primary candidate biocontrol agent is this very tiny little wasp called Alicidia pilicella, which I did my master's degree on. So this wasp oviposits or lays eggs into plant tissue, into the stems, stolons, or leaves of hawkweed plants. And larval feeding diverts nutrients away from plant growth and reproduction. And my colleagues have demonstrated in the lab that this insect does have efficacy in reducing primarily the vegetative output of hawkweed species. And they are plants that are apomictic, and there's many different ploides. So the multiple mode of reproduction, especially in the invaded range, is through clonal reproduction. So looking at an insect that diverts nutrients away from clonal propagation is the most sort of likely to be successful. So when we conduct a classical biological control program, one of the first things we do is look to the literature and identify candidate agents, so in this case, an insect to control a group of invasive weeds. Look in the literature and see, okay, for these weeds and their native range in Europe, what insect species are associated with them? So focusing in on this insect species, the small wasp, there were several mentions in the literature from the early and mid 1900s associating this wasp species with a few different species of hawkweeds in Central Europe. But something that I've learned over time is that, when we find these sorts of records in the literature of an insect species on particular plants, those are largely contributed by entomologists who don't have any background in plant identification and plant taxonomy. So those records may not be correct. Foreshadowing. So the first part of the work that I'll show you today was we wanted to assess what we call the ecological host range of this wasp and its native European range. And what that means really is we wanted to look in its natural environment, which species of hawkweed did it actually use. So in order to do that, we had to learn how to identify hawkweed species, which probably sounds like pretty easy. But actually it's very difficult. But we needed to be able to identify hawkweed species in their native range to be able to accurately assess which are the natural sort of field hosts of this wasp in Europe, and that would give us a good idea about which particular hawkweed species it might be effective against in North America. So we conducted European surveys in the native range of these plant and wasp species. We were based at this institute, CABI in Switzerland. And we traveled around Central Europe, and a lot of our work was based at the Czech Institute of Botany, where there are taxonomists that specialize in this group of plants. They have a hawkweed study group with many researchers that do taxonomy and ecology and lots of different cool things. And so this is where we were first trained at this institute in hawkweed identification by the world experts. So our survey area largely extended between our research institute in Switzerland, where we were based, and sort of satellite areas out of the Czech Institute of Botany. So during this work, Dave and I would go and find populations of hawkweed. We had access to a lot of populations from this hawkweed study group. They could give us hundreds upon hundreds of different sites that we could visit where we could look at different species of hawkweed and survey to see if the gall wasp was associated with them. And we really wanted to maximize the number of hawkweed species we were looking at. Depending on the taxonomic treatment, there could be between 20 and 500 or 600 different species of hawkweeds in Central Europe, depending on if you're a lumper or a splitter. So we also looked at metrics of the plant community because at the time, and I won't present these data, we were interested in abiotic determinants that might be associated with the presence of the gall wasp. Whenever we found plants that were attacked by this gall wasp, so you probably can't really see very well, but here we have little rosettes of hawkweeds and kind of lumpy. Those are all galls along the leaf midrib. So we collect all the infested plants, try and collect the whole specimen to have the taxonomic ID verified, and we did collect some samples for genetic analyses. We also did a lot of surveying of just roadside patches if we would encounter them, and we sort of got, when you're working in a study system, you start to get a sense of where you might want to start looking for something. And so even though we never ended up being able to quantify any sort of metrics of the plant community or the environment associated with the wasps, intuitively we knew where to look for them. Try and develop a model to quantify that, I guess. So we ended up finding this insect primarily in what we considered like 2 general regions or 4 subregions of our survey area in Central Europe. So in this, what I'll call the northern extent of the range, it was really interesting. So we collected plants infested with the gall wasp. We brought them back to the lab, and they were sexual species. There were equal numbers of males and females, and they would undergo 1 generation per year. But the wasps that we collected from the southern extent of the range, there were only females, so they are parthenogenetic line of this wasp, which is relatively common in Hymenoptera, and they had 2 generations per year. So we thought perhaps this is due to geography. I mean, these populations are clearly very separated. The one in the south, there may be just enough degree days for the insect to be able to complete 2 generations per year. But it didn't explain why in the southern part of the range, the wasps were exclusively parthenogenetic, and in the northern extent of the range, we only found sexually reproducing populations. So just summarizing, so sort of these sort of early hints when we were looking at this one wasp that we wanted to control many invasive hawkweeds was that there were differences in the life history in different parts of its distribution. So I'll start to present to you a few different lines of evidence about why this might not just be one species of wasp. So the first, again, is these disjunct life histories in these different parts of the geographic range. So looking a little more carefully in these regions we sampled, so I've just shown you where we found hawkweeds and wasps. So what I didn't tell you, it's not super important, but all the dots are where we found hawkweed populations, and the white bullseyes are where we found the gall wasp. So break this down a little bit. This looks messy, but all you need to look at are the colors of the circles. So in the southern extent of the range, we see all pink circles. And so that represents one species of hawkweed that we largely found and was attacked by this gall-inducing wasp. Whereas from the blue area, the northern range, you see lots of different colors. So we encountered a number of different hawkweed species. And again, these bullseyes represent populations where the wasp was attacking plants. And we see that these bullseyes are on multiple different colors. So the wasp was using multiple different species. But again, thinking about this, it seemed like in the south, the insect really only had access to this pink species. So, is that sort of by chance? It's sort of hard to determine. But what was interesting, sort of coming back to these records that I told you about from the early literature, is, we expected to find the wasp attacking these three species here. Their identity isn't super important. So we weren't surprised when we knocked off these two other species. They hadn't been mentioned in the literature. It wasn't a surprise to us. We didn't find the wasp attacking them. But it was a surprise which species we actually did consistently find the wasp using. And so here I have this pink species boxed in, and that was a species that was mentioned in the literature in a number of sources. So it wasn't a surprise that we had found the wasp using that species. But the species of hawkweeds that I have in the blue and green boxes, those are species that had never been mentioned in the literature as hosts, and yet they were the two most common and almost exclusively used hosts in that blue northern part of the distribution. Whereas these other two species here with these bubbles that represented they'd been mentioned in the literature, we found them either not used or only used at one site and a few gulls on one plant. So our surveys, which had the plant ID verified by taxonomists, showed a different host range than what we would have found if we had just looked at the literature. So really emphasizes the importance of good morphological taxonomic expertise in determining the association between plants and insects or any taxonomic group, especially in something like biological control. So again, coming back to these sort of northern and southern distributions, what I mentioned in the previous slide is, in the south, we only really found this one species of hawkweed, this pink species, and in the north, we found other species being used. So again, we weren't really sure if this is just a pattern associated with geography or if there's another phenomenon. But what these data had started to tell us, first the life history data, now these differences in host use, is that maybe although we were trying to find one insect to control multiple invasive weeds in this complex, we might be dealing with more than one insect, and we might have to look at really matching the right wasp to the right species of plant for its control. So this work, these differences in life history and the ecology of the plant species that they were using led us to thinking that perhaps there might be not just a geographic basis, but a genetic basis to differences in these different populations. And this was sort of motivated by work being done at the time. This was about 10 years ago, and the real onset of DNA barcoding and us understanding that there were cryptic species, particularly of insects, very rampant, and what we thought were one morphological species could often be multiple cryptic genetic species. And so there was a lot of work done at the University of Guelph, of course, but there was also work at this time being done in the field of biological control and understanding that insects that we had previously released that we thought to be one species were in fact actually composed of multiple genetic lines. And to the point now in the literature where there's been a recent review in biological control of weeds, looking at how many instances of this cryptic specialization that we're really dealing with. So coming back, and I meant to have this slide a little bit earlier, but we have these two lines of evidence building, the disjunct life history, different ecological host ranges, which we weren't sure if it was geography or the host plant. We weren't sure how to tease that apart, although we did do some experiments. And we really sort of had this conundrum because we didn't know how to tease apart geography from host plants, because in the south we had the pink species being used, in the north we had the blue and green species being used. And we wanted to investigate the potential for this genetic basis. But then we got really lucky. And we were camping at a site, and this is always like how the good discoveries in biology happen, right? We were camping at this site that we weren't planning on surveying, and we spent the night, and in the morning we wake up and we're washing our dishes in the lake because the tap isn't working, and there's all these matted hawkweed rosettes, and people have been dragging their boats over them, and we're bending down and like, oh, I think there's galls on these plants. So we're like, but this is the pink species, and we found this pink species actually all over this northern distribution. I didn't really show you that, but we never found it attacked. But at this one site where we happened to be washing our dishes, we found all of these galls attacking this pink plant species. So this gave us the data that we needed to really test if this was the patterns, the differences we were seeing in life history and host use were due to geography or had a genetic basis. So we did this just by looking at 3 different gene regions. So barcoding CoA, part of the mitochondrial gene region, and 2 nuclear gene regions. And then we concatenated those 3 DNA sequences and used a Bayesian tool to develop a phylogeny of the different insect populations. So each line that you see on this phylogeny is a different population of the insect that we collected, and the different colors represent the different host plant species. So I've shown you the blue and the green host plant species, the pink host plant species, and I have a couple gray ones in there, which were ones that we collected where we weren't sure of the ID, or some other samples that had been provided to us, and I won't go into details about those. So the first thing we can really see is that we have strong genetic differentiation. We have a branch point of high percentage. Yeah, so you can see the pointer like this large branch point up here that breaks it into the pink and the blue and green host species. So we can clearly see that there is a strong pattern of genetic differentiation. Thanks a lot, sorry. I probably won't use it now. And in the top part of the phylogeny, we have all of the wasps that were collected from our blue and green plant species. And those always came, of course, from the northern extent of the distribution. But in the bottom part of our phylogeny, on one branch, we have all the wasps collected from the pink plant species. And those come from both the southern and northern extent of the range. So this was very clear evidence to us that the basis for this differentiation among these different populations was really based on the host plant and not geography. Because where we had the wasps from the pink site, from the northern extent of the range, they didn't group with their neighbors. We collected those wasps at the same site and at nearby sites as to wasps using the blue and green species. And if there had been a geographic basis, then we would have found those populations clustering in the top. Instead, we found them to cluster with other wasp populations from the same host plant, but from different part of the geographic range. So when we come back to these different lines of evidence, the disjunct life histories we thought might be due to geography. The divergent ecological host ranges, we weren't sure if it was geography or host plant until we had that one population. And when we looked at the patterns of genetic differentiation, we saw that it was clearly a host or plant-driven pattern and not a geographic pattern. So very clearly from this work, we started testing further hypotheses about the role of the host plant in mediating these different life histories and host ranges. And I won't present work on this today, but some collaborators of mine at Lethbridge, they've done a number of follow-up experiments and have tried to sort of take wasps from one host plant species and move them to the other. They won't attack it. They've tried to induce changing the volatilism and see if we can get the one that produces only 1 generation per year to produce 2 generations per year. If they have increased number of degree days, they won't budge. So these are very fixed biological differences in these two cryptic host associated lineages of this one wasp species. So when it comes back to, not just thinking about the cool biology of the system, but thinking of these insects as tools, as potential biocontrol agents, it's then really essential that we're matching the right line of the wasp to the right plant target. So the second part of the work I'll present to you today is when we came back from Europe and had collected these data and done these analyses and realized we needed to do a lot of work on the ground here in BC and better identify the hawkweed species on the landscape so that we would know where to release each of these 2 different wasp biotypes if they would be approved. So the work that Dave and I did, funded largely by FLNRO, was to clarify and describe the distribution of the different invasive hawkweed species in BC and identify some candidate release sites. So just coming back to these pretty pictures of hawkweed species. In terms of identifying these species on the landscape, well, one has orange flowers, so that one's pretty easy. As long as the plants are in flower, we don't have to worry about it too much. It's rarely misidentified. This species here, which is actually the pink species from our study, is undergoing a management practice called early detection rapid response. So it's actually very rare that we find populations of this plant in British Columbia. And if we do, it's reported and it's removed. But when we're looking at the remainder of species, hawkweed species in BC, so the blue and the green species, I only showed it in blue, but are these 2 over here. This was the blue and this is the green, but it's still blue. And this was the pink one. Well, what's interesting is this species of hawkweed here is actually a hybrid. So these plants also, they're not just polyploids, they're not just apomictic, they also hybridize like crazy. It's a hybrid offspring of this species. So we had good reason to believe, and it was confirmed in laboratory tests, that it would be susceptible to attack by the pink lineage of the wasp. So we really needed to be able to identify on the landscape in BC which species of hawkweeds were occurring where, because there'd be no use in releasing either of these wasp lines on populations of these hawkweed species or this hawkweed species, and there'd be no use in releasing the pink wasp on populations of plants that only the blue wasp would accept. So this is just a little bit about the taxonomy and hybridization of these species. So really there's 2 basic species of the yellow-flowered hawkweeds that we have in British Columbia, and then subspecies or different hybrid species. And so when we're conducting identifications, this is just sort of to give you a bit of context about why it might be so difficult to identify these different species, is that they're very closely related, but they actually do occupy different ecological niches. So they often can co-occur, but there's a paper from, I believe, like the mid-90s that describes the ecological niches of a few of these species, and while they overlap, there are differences. So Dave and I set out to survey the hawkweed species in sort of central BC, and I showed you that map before of all those purple dots of what were yellow-flowered hawkweed species. Even though there have been previous efforts in the province to ID hawkweeds to species, it had been done at certain sites, and there hadn't been a huge volume of data collected. So, really we only knew in a large sense where the yellow hawkweed species were and not necessarily what they were. So we spent 2 summers driving and surveying hawkweeds across sort of central and eastern BC. This involved going to lots of different types of sites and lots of very cool areas and finding patches of hawkweeds. And in order to cover as much ground as possible, we weren't always surveying when plants were in flower. So we ended up surveying, I think, over 3,000 sites. And so just the main message to get here is that the dots on the map are no longer one color. So we've been able to resolve the identity of different hawkweed species at thousands of different sites across BC in preparation for biological control. So these data just sort of look at how many sites of each species that we ended up dealing with, and maybe I was exaggerating before. It felt like 3,000, maybe it was close to 1,400. 4,000, 6,000, 10,000. Every time I'll tell the story, it'll get to be a bigger number. But something that emphasized here is that so the numbers in parentheses were the numbers of sites we visited that were supposed to have that species. And then the number not in parentheses is where we actually found that species. So in 2011, when we were surveying in the southern part of BC, there were only 8 sites that were supposed to have this one species, and we found it at 171 sites. But then in 2012, when we surveyed the northern part of the province, there were 18 sites that were supposed to have it, and those were mostly mis-ID'd because we only found that species at 2 of those sites. So it sort of went in both directions, where we would go to a site and we'd most often find species that hadn't previously been recorded, but there had also been a number of misidentifications that we corrected. So these maps just summarize sort of the differences in hawkweed distributions before and after our different surveys. So on the left, we have before, and whereas the map I showed you before was purple dots, these are mostly yellow pies, and this is by forest district using the forest districts at the time that we did the surveys. So primarily at the time before we started our surveys, in yellow here is just what were considered yellow flowered hawkweed species, and so in each forest district, the majority of records were just for yellow flowered hawkweed species. The gray, actually, this is a synonym for yellow hawkweed, and so yellow and gray are basically just yellow. So almost the whole pie is just yellow hawkweed. We don't know what species it is. So after our surveying, we identified particular species at sites and then could analyze the data by forest district and look at the distribution of the different hawkweed species throughout the province. And again, it's really just, we can see with the colors, we don't need to worry too much about the particular species, but of course the distribution of these species on the landscape is not uniform. It differs likely based on the time and location of initial invasion of these different species and how they've been transported, but maybe slightly based on the different ecological niches also. So coming back to biological control, where we found populations of what were the blue and green plant species, and of course the colors are different on here, and I couldn't go back and change like all of the different colors. But on these maps, those colors are this dark red and the orange. So this blue now goes to dark orange and red, and so we would see that it would make sense to release this lineage of the wasp at sites primarily in this region, but also on this species represented by the red in the southern extent of the range. And that's where in the previous data that I showed you, where we had uncovered many more records, like the 171 records, we had uncovered a lot of records of this particular hawkweed species in that region. Whereas the wasp lineage that I've shown you throughout the presentation in pink, there's actually relatively few areas in the province where we would probably want to release it. The species that it would be targeting would be this bright red species here, and we have population here and a population here. There are a few other smaller populations through the province, but its range is relatively restricted. So sort of, I don't have slides for this, but bringing it back before we had done this work, the initial populations where this wasp had been collected were around CABI, that institute in Switzerland where we had been working. And so there had been populations of this wasp collected there and kept in rearing, and that had been what the initial testing for this species had been conducted with. So when my collaborators were looking at sort of the efficacy and specificity of this species of wasp, they had been using this wasp biotype because it was very close at hand. And until we had done broader geographic surveys and uncovered these different patterns in life history, host use, and the genetic basis, we wouldn't have necessarily known that this species of wasp is not going to be effective on the majority of hawkweeds in BC. That we had to look to not northern Europe, but the northern extent of the range to find this different lineage of the wasp that is specific to these hawkweed species that are very broadly distributed in British Columbia. So I think I went a little bit fast. That was the end of what I was planning on talking to you guys about.
Thank you.
Thank you, Chandra. Time for questions.
They asked me a lot earlier, so.
Have you released any of these yet?
No, so they haven't been approved yet. So what's interesting, and now that I know I would have had time, so when I was doing the genetic work, you obtain DNA sequences like you do in your lab, you blast them in GenBank and look for the most, closely related species or individuals. And we actually found that the species is already present in North America. So it's present in Ontario, and we've subsequently had collaborators go and find that it is attacking hawkweed plants in Ontario, but it's the pink lineage of the wasp that's present. And so we're kind of into a new territory in biological control, and it's not specific to this study system. It's happening in all kinds of study systems where one type might be inventive, but the populations that we actually want to introduce are not yet here, but they're considered the same species. So does this speed up the process of getting permission to release what we, if we didn't know about the genetic work, we would think, oh, this species is already present in Canada, like we don't have to worry as much, it's already inventive. But we do know that they're two different lineages, and even if they're the same species, they're very clearly of different biologies. So that's just extra information. It's not the answer to your question, but I have time to talk about that.
But it will complicate things because it could be perceived as gray.
Yeah. So there's a similar case happening right now with a parasitoid for brown marmorated stink bug. So there's one population that's been detected in Ontario, but it's not the same genetic line that the what we call host range testing. So looking at the potential impact on native species, it's a different population. So we get into this gray area, and it's happening in a lot of study systems.
Two things. One, thanks for the talk, and if I could just ask you to step back into the little.
I broke mine. Yeah.
Thank you. I'm following along here. Upstairs, we've been talking about kind of the spread of the population of this, and you mentioned you found lots of these species wherever you found recreationalists.
The weeds, yeah.
The weeds specifically. Are there any kind of partner studies going on, say with tourism management or ranching or whatever, where there's kind of overlays of data? Because that'd be really interesting, where people are tracking, say, movement patterns of recreationalists along with this strategy.
Yeah, Susan might be able to answer that question better than me. I don't know, like when we did this type of survey work, we when you're surveying invasive weeds for the province of BC, you're always reporting the jurisdiction to the best of your knowledge. And so like I said, we often find invasive weeds along major transportation corridors, under power lines, at forest rec sites. So there's a lot of jurisdictions involved, but I haven't been working in this line of work for the last 7 years. I'm just kind of coming back to it, so I'm not sure what kinds of partnerships exist, but the data is there to sort of identify who the players are, I would say. Do you want to add anything?
I'm with transportation. Yeah, so the bees also. In our database, we do have quite a few species mapped. Historically, before a lot of work that Chandra did, as you see, that they were mixed together with the yellows. So, except for the sites that they went to, there's still a lot of unknown, misidentified plants there, but they do group some of them. Like the one plant species that she mentioned, Mosier hawkweed, is
The pink one.
the pink one, is in 2 parks. Or sorry, one main park over in Nelson area, and that one's actively being controlled. They thought there was another infestation, but it turned out to be whiplash, which is the hybrid of that one. And whiplash grows quite clumpy, but it doesn't seem to show a pattern. It's in certain areas, but some of the other species, I don't know if it's actually especially the orange. It's
Rail lines are another, like in northern BC, we often found them pull over at the, you'd be surveying and like rangeland and you might find like 1 or 2 species of hawkweed. If you go to the rail line, you find 7.
There's so many vectors. Yes, there are things that are moving them, but there's so many vectors that pick any one.
So you mentioned that the hawkweeds grow in really high densities, and I think one of the previous speakers on invasive species mentioned that sometimes biocontrol agents aren't as effective when plants are growing in very high density because they can't effectively reduce reproduction enough to have a significant effect on the population. So, like, how are the wasps doing that?
That's a super great question. So our collaborator in Lethbridge, no, it's like really relevant to the study system. So our collaborator in Lethbridge has been looking at that and trying to determine like a problem with the previous sort of efficacy work had been like putting a hawkweed plant in a pot and then seeing if the wasp was really efficacious at controlling it. Well, in a pot, these plants like go crazy, whereas in a population, they're extremely crowded. And so she's been looking at like in sort of like different densities of crowding, like can it slow the expansion of that patch? And that's what she's found is that the overall reduction in vigor slows the spread of her like experimental patch. So I think like for a clonal species like hawkweeds or other clonal species, I think it's more about slowing the spread and then like, so slowing the spread of those patches probably, then like maybe getting a great reduction like on the landscape in that particular area.
And then maybe use other measures to actually remove the species at that point once you slow the
Yeah,
spread.
they're so ubiquitous across the landscape. Like if we could use mechanical or chemical methods, we would. Biological control, we've been working like we have been working on hawkweed biocontrol for like, I want to say almost 20 years, like 50, like, yeah. So, and we have 2 other species of insects that have been approved for leaves. They have a bit of like different host range and I'm starting to spiral.
So Cole was referring to Jennifer Williams' talk, and she was also looking at the rate of evolutionary change in association with spread. And it sort of makes you think that, the hawkweed that has been introduced to BC, it's been here for how many years?
Like over 100, most of them, we think.
And often that evolutionary change, or at least Jennifer
Yeah,
determined
happens
that
quickly.
revolutionary change can happen very quickly. Maybe our semi-naturalized hawkweed are becoming so different from the hawkweed in Europe and the bugs that are on those hawkweed that there's going to be a problem with that association.
So those are really great questions. And I think those are real issues that we're dealing with in biological control of weeds and other invasive species. In this study system, I think it's less of a problem. And a few reasons I think that. So like I said, they largely reproduce asexually. So like the frequency of genotypes, like we have, of course, the populations are evolving because we have different frequencies of genotypes on the landscape that have changed here over time. I'm sure of that. But we, in some work that I thought I didn't have time to get to, but have slides for, we did a study where we compared the genetic fingerprint using AFLPs of hawkweed populations in BC and in Central Europe, and we found some of the same exact genotypes. So I think because they're asexual, like we see fixation and we see some of the same genotypes from Europe present here, but we also saw genotypes in BC that we weren't able to recover in Europe. So whether those are novel genotypes or genotypes we were able to sample. I think in terms of like the interaction with the insect, so when we do like the testing on the small scale, like in quarantine, we use plants that are collected from here. But these, the plants have had time to adapt. They're much more vigorous and invasive than their conspecifics in the native range. there likely will be a different interaction between the biocontrol agent. And we hope that that's an interaction that, by the biocontrol agent feeding, the plant has to now divert resources towards plant defense. And that's one of the major tenets of biological control, of course, is that, plants stop producing those defenses. They can allocate resources to competitive ability, to other metrics, and that by reuniting them with the insect and having to allocate defenses, that they stop putting all their energy towards that. So I think it's a real issue in a lot of systems. That maybe I'm naive, but I think it's less of an issue in this system because they're clonal. That's a really good question. Thank you.
So it seems like a lot of the research is looking at plant host specificity.
Totally.
How much research goes into fulfilling or looking at the other ecological roles of the biocontrol? So like interactions between that biocontrol and other invertebrates, or of them becoming like a prey species of insect or
close to zero, I would say. So that's not part of the testing generally that we do in biological control programs, but it's changing a bit in biocontrol of insects. And I think it's just because we think about insects differently than we think about plants. Like plants are, we think of them as being fixed on the landscape, and we know insects are moving around. So I think we tend to think about the community interactions more with insects. And so if we're considering introducing, for insects, we usually introduce a small wasp as well. It's called a parasitoid, and it would lay its egg into developing insect. We think about, if we release a wasp to control a stink bug, we know there's other wasps that prey on other wasps. They're called hyperparasitoids. Like, we start looking at those interactions more. But that's more in it's not the right word, but like academic sense. Like, it's more for the interest in the ecology rather than like anything to do with legislation or permitting. Like, we don't look at other ecological interactions, I think, because we don't have the resources really to do that. But for sure, if we introduce a new community member, and especially if they become like, we've had very successful biological control of weeds where we might release an insect and the population numbers like, boom, and they distribute. Well, that's a food source for birds, for rodents, like for other insects. It's going to have community level impacts for sure, but we don't really measure those.
Any more questions? All right, well, thank you. Oh, there's my
Just back to the hawkweed. So where you're seeing it distributed is where you've seen relatively permanent disturbance on the landscape. So railroad tracks, areas subjected to ranching.
Yeah,
It's not biased by the fact that that's where you can get to easiest. Would you expect to say sometimes find it in pristine alpine locations?
We did, so they're not shade tolerant. So like wherever the canopy cover was thick enough, like we wouldn't find them. So in terms of the alpine, I would say we didn't specifically really sample. Like, we were definitely limited in terms of like, we didn't ever hike into a site to look for hawkweed species. But in other work that we did in similar areas, like the work that we did collaborating with Law, like we did go into alpine areas and we only ever found native hawkweeds, and that's a small sample size, but it's interesting. I like the way you're thinking about it, because in Central Europe, like we find these species in the, like, it's not technically the alpine, but like in the high elevation, like montane meadows, that's their native distribution. But in Europe, those are highly disturbed environments, whereas our alpine meadows are presumably a lot less disturbed. And in Europe, those alpine meadows have been subject to grazing for, I don't know, hundreds of years. But yes, our sampling is biased in terms of like going to disturbed sites and accessible sites, but we did see it like in like fairly new plantations of like in forest regeneration, where until the canopy is going to close in, like I had one picture where like the whole understory was just hawkweeds. There's no native vegetation.
Thanks.
Yeah, in northern BC, we see hawkweeds wherever anybody has ever walked. Anyone.
In northern BC.
In northern BC, and there's the only place where we don't find hawkweeds, because we've been trying to keep them out of the area is in the Peace River, where we're trying to keep it out of agriculture areas. But hawkweed has been transported up to Pink Mountain, partway to Fort Nelson, and we've got whiplash up Watson Lake in the far northwestern BC. It's marching its way north. Anywhere that you have people moving, both yellow and orange.
And I mean that like it's not a surprise considering its distribution in Central Europe and the habitats it inhabits there. Presumably, like in the less disturbed areas, I think there's just enough like resilience, like that hawkweeds aren't, if they're not getting access, or there's not that disturbed niche to inhabit.
Has it made it to the Yukon?
I don't know.
They have a whole herbarium in White Horse.
Cool. Do they need them identified?
Right then. Well, thanks so
Thanks,
much.
Tom.
Yeah. Show.
I have one more thing. My pitch that I was supposed to do. A pitch. So you like ecology, evolution. I don't think there's many bug people, but the next meeting of the Canadian Society for Ecology and Evolution is in Fredericton, New Brunswick in August. I'm one of the chairs of the meeting. It'll be awesome. Please come.
That's true. Hey, can you ask somebody the mantis question? Email me. Ask somebody the mantis question.
Yeah.